So thank you very much, Simon, for the introduction and for giving me the opportunity to present my work here today. So as Simon said, I will be talking to you about the use of DNA nanotechnology and how we can use DNA nanostructures to target specific bacteria while simultaneously delivering active antimicrobials to these bacteria. And of course, the first question is, why do we care? Why do we want to do this? Why do we want to have sophisticated delivery systems that target bacteria? And I'm sure that this audience doesn't really need convincing, but of course the answer to that is the emergence of antibiotic resistance. So as you probably already know, it is predicted that by 2050, deaths caused by antimicrobial resistant infections are set to overcome deaths caused by cancer and diabetes combined. And these numbers were actually reviewed very recently for the wars. This prediction, of course, is true if nothing changes in the ways that we currently address antimicrobial resistant infections. And luckily, there is a lot of work being done in that direction, broadly categorized in three main categories. The first is new antibiotic development. The second is the use of biophysical and biochemical approaches so that we better understand bacterial pathogens and their strengths and vulnerabilities. And the third one has to do with inventing ways with which we can deliver antimicrobials in a more targeted, more specific way, and therefore potentiate these antimicrobials and hopefully give them a new lease of life. And this is where I will be focusing my talk today. So there is already a lot of interesting science going on in terms of antimicrobial delivery, and it's mostly focused around nanotechnology. And there is a huge array of techniques already investigated, starting from dendrimers, carbon nanotubes, MOFs, all sorts of nanoparticles, ranging from the completely inorganic metallic ones to the fully biomimetic ones. And if you like, the weakness of this approach is the lack of targeted delivery, of the lack of specific delivery. And this is what we are trying to do through the use of DNA nanotechnology. So a DNA nanostructure is formed by folding, by taking a long single-stranded circular piece of DNA, which is folded and held into place through the use of hundreds of short oligonucleotides that we call staples. We can design and synthesize pretty much any 2D or 3D shape that we can imagine. And on top of that, we can functionalize our staple oligonucleotides to carry active antimicrobials or active molecules of any sort. So this gives us a highly customizable and very flexible platform to fine-tune according to our specific applications. So the nanostructures that we are using look like this one that you can see on the screen. So they are a rectangle that has 5 holes, 5 wells in the middle, and looks a little bit like the face of a 5 on a dice. And we have assigned 3 different functionalities to these nanostructures. The first one is that we have a biotin in each one of the wells, and through a streptavidin bridge, we are attaching to those biotins biotinylated lysozyme, which is our active antimicrobial in this case. The second one is the addition of 16 aptamers in the perimeter of the nanostructure that have the ability to bind both to E. coli and to Bacillus subtilis. And the last one is the addition of 4 Alexa 647 molecules that will act as detection beacons for these nanostructures. So once we have designed and synthesized their nanostructure, we wanted to test each one of these functionalities that we have attributed to them. So to begin with, we just tested the successful formation of the nanostructures. And you can see here on the top row, a 2D and 3D representation of those. And we found that they form as planned and with an efficiency of 92%. So we then went on to incubate the nanostructures with streptavidin, and you can see in the second row the streptavidin molecules appearing as blobs on the sides of the wells here. And as a last step, we incubated those nanostructures with lysozyme, and at the bottom row, you can see the fully loaded nanostructures, both with streptavidin and biotinylated lysozyme. And we also performed the volume analysis on those blobs in the wells, and we saw a shift of the volume between the just streptavidin incubated ones and the ones that were also carrying the lysozyme. And that corresponded to approximately 1 or 2 lysozyme molecules per well per streptavidin. So that was encouraging, but of course the second step would be to test whether these nanostructures actually do interact with the target bacteria. And we performed some structured elimination microscopy to understand that. And what we saw was indeed a decent association of our nanostructures that you can see here in magenta with the target bacteria that you can see in green. And the coverage of the surface of the bacterium was on average 20% for either type of bacterium. We see differences in the ways that the nanostructures seem to interact with the target bacteria, and we think that this might be because of the differences on the surface between a gram-negative and a gram-positive bacterium. So again, that was good news, but of course, the main question is there any point in this? Do we potentiate at all these antimicrobials through this targeted delivery in comparison to just putting the lysozyme in solution like we would normally do? So we did some growth curve analysis to understand that, and what we saw was that in both cases, we see a reduction in the growth rate of the bacteria, but also a reduction in the amount of total biomass that we would have in these cultures. When we further quantify the growth rates for these bacteria, we also see that in the case of E. coli, we get a reduction in the growth rate through adding just plain nanostructures. And by plain, I mean nanostructures that do have the aptamers on their perimeter, but they're not carrying any antimicrobial. And this is where we're at the minute. We have these promising results, but what we want to go further and understand is how do these nanostructures interact with the bacterial surface? Where exactly do these aptamers bind? Because the aptamers that we're using are a bit of a black box. We don't know where exactly they are. And what happens downstream in the bacterium once this binding has taken place? And my colleague Karen Bali is giving a flash presentation later on to talk you through one of the ways that we're taking to understand this. But in the last few minutes that I've got, I want to show you a slight side direction that we have taken in the last couple of months that is giving us some interesting results. And this work is led by a very talented PhD student in the lab, Ioanna. So what Anna is doing is she has designed triangular nanostructures that still have the aptamers that can bind the bacterial surface, but at the same time they have sticky ends so that they would be allowed to hybridize, like you can see here on the left of the screen. And again, we did our usual tests of the nanostructures. So we tested their flexibility and mechanical stability in fluid through computation and their successful formation through AFM. And once we were satisfied with this, we went on further to attach the sticky ends to cause them to hybridize. And if we take a closer look here on this image on the right, we can see in this zoomed in image, for example, 9 of the 10 hybridization points haven't formed successfully. Of course, as you can tell, the arrays are not yet perfect, so they have holes or we have mismatched assemblies. Yep, but we are getting towards full arrays, and in the meantime, we just went and had a quick look at what these arrays might be doing to bacterial targets. And again, through structured elimination microscopy, we are seeing a few differences between the arrays and the individual nanostructures. So what we see is these larger assemblies that are kind of enveloping a number of bacteria rather than individual decoration of bacteria. And when we have a look through TEM, and this is very, very fresh data from just last week, we see normal-ish looking E. coli without the DNA origami arrays, and then this damage perhaps on the bacteria surface when these arrays are added. So just to summarize, in the interest of time, we know that we can make DNA nanostructures, we can design them so that they can be functionalized to specifically target bacteria while carrying active antimicrobials, and the targeted localized delivery of these antimicrobials potentiates them. And we can also use similar DNA nanostructures that can self-assemble into large nanometer arrays. And our preliminary data show that these arrays can interact with bacterial targets successfully. So with this, I'd like to leave this here. Thank you, all of you, for your attention, all the group members and our collaborators, and of course, our funders. And I'd be happy to take any questions.
Thank you very much, Ioanna. That was great, beautiful images. We've got a time for a couple of questions. We've already got a couple in the chat. So first of all, do you believe that the bacteria will develop resistance to your aptamers, and how long are the aptamer extensions?
This is an excellent question. I mean, the immediate answer is I don't know, because we don't know exactly where the aptamers are binding right now. So these aptamers are developed against whole bacteria, against whole cells. So we only know whether they bind or not bind. So if they're binding on a poring, or if they're binding just on LPS, or if they're binding in any specific part of the bacterial surface, that will probably answer more precisely whether or how quickly or whether a resistance will be developed. And that is what we really, really want to understand now. This is my next step, to try and develop aptamers for specific parts of the bacterial surface, and then test the binding of those aptamers and the downstream effects in a much more precise way. The aptamers that we're using are 45 bases long.
Okay,
So
and then thank you. Then the next question was, on your images with Bacillus subtilis and E. coli, your fluorescent images, there were some vesicles or round structures associated with Bacillus. What were those? Were those live cells or spores? Or
so it's an excellent question. What we think these are is mini cells. So bacteria are known to produce these mini cells on their ends. It's not entirely known what they do. There is this theory that they are kind of garbage disposal mechanisms, so they contain damaged parts of the bacterium. And that's what we think they are, because they are membranous. They don't contain DNA as far as we've seen, but they are there, and we've seen them much more in the case of the gram positive than in the gram negative. So this is our current speculation, I would say, that these are mini cells, but we haven't tested this hypothesis.
Okay, that's great. You've answered the next questions. The last two I'm going to join together, which was the DNA that you use to do your origami. Does it have a biological function? Does it get inside the bacteria, and can they use it as a nutrient?
This is a very good question. So the DNA nanostructure as such, of course, it doesn't have another biological function. The backbone DNA is extracted from virus, so it's a viral single-stranded DNA. But once it's folded, it's almost inert, if you like. So eventually the nanostructures will start getting eaten up by DNases and so on. And each nanostructure, depending on its shape, has different weak points. But I would be very surprised if this DNA assumed a functional role ever. So whether they can be inserted in the bacteria, it depends between gram-negative and gram-positive. So DNA nanostructures have been seen to get endocytosed in mammalian cells, but of course bacteria don't endocytose in the same way. My feeling, if you like, is that the nanostructures would need some help to get into the bacteria. So whether this is covering in liposomes that can then kind of fuse with the bacterial membrane, more so in gram-positive cases and so on. But this is something we are really trying to do at the minute, to try and get them into bacteria.
That's brilliant. Well, thank you very much.